Okay, so we'll start now the last part of this presentation, and I want to talk about the nanoparticle from combustion and the interaction with biological systems. So here I have a cartoon of one nanoparticle from the SNAPS code I showed you before, and the idea is to see how they interact with the biological systems like cellular membrane. Before doing that, I just have a couple of slides about some of the effect, and this is a little bit of history. So in 1775, Percival Pott, that was an English physician, started noticing that the majority of men of this patient that were male basically had some kind of cancer, and they were all chimney sweeps basically. And so this kind of study led to a parallel act of the parliament preventing the men under the age of 16 from becoming chimney sweeps, and forbidding men under 21 years old from entering a chimney. So there was already some correlation between the effect that soot could have and the health effect. In addition, if you look, for example, at tobacco and the species that are produced, some of those are very harmful chemicals with carcinogenic properties. There is some research that reports that the people who smoke, basically they have to sustain one genetic mutation for every 15 cigarettes they smoke. And so they found that for the entire genome of a lung cancer cell, they found almost 23,000 mutations in smokers. So that led me to the interest in the risk assessment of environmental nanoparticle. And if you want more like the safety of this material. And so when we talk about risk assessment, there are two major components that we need to take into account. It is the hazard and the exposure. So if you think about that, yes, maybe nanotubes can have a problem, can cause trouble to your body, but how much are you exposed to nanotubes every day? I mean, unless you work in a lab with the nanotubes, you don't see nanotubes in this room, for example. But what is the problem is that, so this is negligible, but instead we are exposed every day to, for example, whatever comes out of an engine of a car if you just walk outside in some ways. And so in order to, I got interested in risk assessment because now I had some kind of way to describe a nanoparticle from a combustion source. The reason why nanoparticles are also very important, and the reason why they are concerned for the health here is showing that as a function of the diameter, how much is the deposition in the body. So this is telling you that this is the total, but the smaller the particle, the higher is the concentration that gets into the body. So this is the lung, for example. And so the smaller, the worse, if you want. So the title I had here is Why Nanoparticles Are Bad and Therefore Important. The graph on the right shows something similar. So this is telling you the diameter of the nanoparticle from the combustion, for example, as a function of the number of particles per day in your lung. And so the smaller the particles here, so 0.1, the higher is the number that are deposited every day in your lung, for example. And so what we worry about, or what we should take into account, is the formation of the nanoparticle. So going back to your question, in which, yes, we are looking for a model to suit, but also, again, very interested in this transition to nanoparticle for a series of applications, and this is one of them. And so what you find in the literature usually is what they call epidemiological studies. So they have, for example, usually rats, mice, and they expose them to pollutant from an engine. For example, they have a chamber in which these poor mice, they breathe, everything comes out of an engine. And so they have a correlation between exposure and symptoms, right? They can have asthma, they can have increased mortality, they have seen damage to DNA, they have seen damage to cytokine, different things in the body. But what we are trying to do with the molecular dynamics with this regard was trying to bridge those two evidence, if you want. And so can molecular dynamics tell me something about how a nanoparticle, for example, from combustion will interact with a membrane, with a cell in the body? And so the idea was that also what is the size, what is the makeup of those nanoparticles, we want to know these things. So if you want to talk about the biological system, and again, this is, I'm not telling you a lot, but the cells. So the body has a lot of cells, okay? And they are very different, but they all contain 3 parts that are very similar. And one of those is the cell membrane. So things are outside, they have to cross the membrane to get into the cell. And so one thing that I was interesting is that if I have a nanoparticle outside my cell, this nanoparticle will go through the membrane inside the cell, yes or not. And so how can we do this with molecular dynamics? This is a huge system. So molecular dynamics, remember, is very small. And so we took like a small volume here in the membrane and translate this into a system that molecular dynamics can handle. And so the membrane becomes something that, like this. So a membrane is what they call a lipid bilayer. So it's lipid, basically. You have those are called heads that are phosphor, nitrogen, some chemistry, and then they have these orange tails that are carbon and hydrogen. This is the membrane. So this is outside the membrane. This is what they cross before getting into the cell. And the other component of this membrane is cholesterol that many of you, I'm sure, are familiar with. So, you know, they usually measure the cholesterol. Cholesterol is always in the membrane of the cells. It does some flexibility. If it's too much, too dense, it's usually also a problem. So the way the cholesterol is a molecule that looks like this. And the reason why I want to show this is because as an aromatic, so something like a combustion that you are familiar with, the 6-member rings, not that different. And then it has also an OH, and then it has some kind of aliphatic chain here, okay? And the way the cholesterol positions itself into the membrane is like this. And the reason is because OH is hydrophilic. He likes water, and so he's outside. While instead, this other part, like an aromatic in a combustion, he likes the tails that are carbon and hydrogen as well. So hydrophobic, they don't like water, they stay inside, are the tails and the cholesterol. OH likes water, so he's outside this way. So that's the way usually he sits in the membrane. Now, we translate this into a membrane, the molecular dynamics, and you start seeing a box that looks like this. So the small dots here is water, and this one are, this is the lipid bilayer. So the first leaflet and the mirror image at the bottom. This one is not the green head, but I have to spell out all my chemistry. So I have nitrogen, phosphorus here, and then I have those green ones that are the tails that I was talking about. And then darker green, I'm not sure how much you see, is the cholesterol that is in between. So I go from the pictures on the left, I try to atomistically translate into these ones. And so the question that we were thinking is, you know, what is what kind of, so when you expose a cell to a nanoparticle, for example, will they go through? And what are the characteristics of this nanoparticle that you are interested? So a molecular dynamic simulations, this is the lipid bilayer I just mentioned, and now I color-coded a little bit different so it's easier to see. And again, the darker green, blue is the cholesterol. So the nanoparticle I show you from combustion is weird. It's not necessarily a sphere like a Fullerene or a nanotubes, but it can have a different configuration. And so what molecular dynamics does is something that looks like this. You wait over time, and then you start seeing if, for example, this conformation, this chemistry, this morphology will go through or not in the membrane, okay? And so, similar to what you learned from combustion yesterday, we look at the free energy. So this graph is showing you, this is the membrane. 0 is the center of the membrane, and 3 is basically outside, where you have water and you have all the particles. And this graph is showing you the free energy that you, by now, have a feeling on how to compute with MD. And this is telling you that as soon as, for example, the round-shaped nanoparticles gets close to the membrane, it will jump in. There is no barrier to get to this one. So if this nanoparticle gets close outside to the membrane, the free energy to go in, it will be negative. So it's a spontaneously process will go in. The reason why you see a minimum here is because you are in the tails, and if remember I told you the tails are carbon and hydrogen, so they like another carbon and hydrogen, so they will go here. It's a balance between the H enthalpy and the entropy, so the space that they have in the tails, but this nanoparticle will end up here. So we work with the people who are doing some biological experiments. So they took, they bought like a fullerant, so something that we could model, characterize easily, and then they grew some cells, like the body cells, there's a liver cells actually, and what they did is dump those fullerants, so carbonaceous material. Remember that fullerants are produced in flames also, okay? So you can find them as one of the byproducts of nanoparticles. And so what they saw is that the nanoparticles will, here what you see a TM, so micrograph of those cells inside the cells. So somehow the C60, the nanoparticle, the carbonaceous nanoparticle with those experiments will go through. They found it inside. They found it inside the cells, closer to the nuclei and other parts of the cell. So experiments were kind of used to help the MD, but also to guide the evidence that what we saw is basically the nanoparticle going through the systems. But of course, when you run a simulation, when you run an experiment, you don't have one molecule, right? You have a lot, you have a cluster of nanoparticles, like in combustion. And so we have additional simulation that we were trying to look, understanding how things were working, if you will. So we took a big cluster of nanoparticles that were all the C60 here, and then we look at interaction with the membrane that I just showed you. And this movie, what this basically is color-coded in terms of height. So what that means is that as soon as the membrane starts seeing this blob of nanoparticle coming in, some of the lipids will start climbing over this blob. And so you will see the white and the blue going higher. Eventually, the blob will go through the system, basically. And so how do we also validate those kind of simulations? Was with experiment. So my MD will take the nanoparticle and will see that will go through the membrane. And this is an experiment that was done with NMR. But the point is to show you here in a simple way that this is the order of the tails in the membrane. So if you don't have any nanoparticles, this is what you should see, basically. In the moment we added the nanoparticle, what we saw is that the order, so the way the legs were basically in the membrane, will change dramatically. And they changed around, this is the carbon in the tail, carbon 8, that correspond with the minimum that I show you in the free energy. So the free energy was telling my nanoparticle will stop in that kind of dip that correspond to one of the chains in the membrane. So there was a validation. So we started from the real cell. Then we did some of those experiments with NMR. And then the MD, basically we put the three together and they were able to MD validate. We validated with the experiment. The other thing that you can do is this one. And I think this might be one of the last things that I have. So we were very much interested in looking at those protocols to define the health effect of nanomaterials. And so there was an article a few years ago in which they were defining the health effect of the nanoparticle in the range up to 2-3 nanometers, similar to what the fullerene does in terms of damage. So the idea is that if you have another organic nanoparticle that has a similar size of the fullerene, will cause the same damage that fullerene does. Okay, so that was whatever. And so we ran a molecular dynamic simulations for this. And again, now you know as a membrane, so it is a lipid bilayer again, and we took 4 different nanoparticles. So one was the fullerene. The second one was an open fullerene, and the reason why we did that is because we wanted to not change the mass, but we wanted to change the exposed area of those nanoparticles. So the idea is that if you're interested in the health effect, what parameters in the nanoparticle you want to take into account for that? And then we compare with the combustion product of the similar size of the nanoparticle, but they were coming from different flames. And so the difference between the third one and the fourth is this is hydrophobic, so it's a carbon and hydrogen, and this one was hydrophilic, so you had some kind of oxygenated species we talked about this today, right? There is oxygen sometimes in the nanoparticles. So we look at molecular dynamic simulations, and the question we ask is, how can we use this to understand something about the mechanism? And this is again a free energy that you have a feeling for at this point. So similar to what I showed you before, 0 is the center of the membrane, 3 is outside. So all the nanoparticles are outside initially, and what this graph is showing you as a free energy. So C60 is what you saw before as a black. The open C60, that this guy here, is the green. So the different result tells you that mass is not a discriminant. So if you want to say particle A and particle B have a different health effect, mass does not distinguish them, but you need something more. Because these two, they have the same mass, the black and the green, but the tendency to get into the membrane is completely different. So maybe exposed area is something else that we want to consider in the health effect. And then the other two is the blue and the red. The blue is the combustion nanoparticles, carbon and hydrogen, and what you see is that the free energy is even lower. So somehow the fact that it has a different shape and has a carbon and hydrogen ratio brings you this value down in the system. And again, the free energy is basically a combination of the entropy and the enthalpy of the system also. And the last one is the red one. Red one, you can see this curve is much higher, and the reason why is that is because it has oxygen on the top. So what that means is that it's hydrophilic, he likes water. And so if you remember the tails that start usually between 2 and go to 0 in the center of the membrane, they are hydrophobic, so they don't like each other. And so basically you will not see the same dip that you see in the other cases. But you will see that the red will go down immediately because the first part is hydrophilic also. So a lot of information, but the point is, first of all, you can use MD to do something else, not only the combustion stuff. The second part is that what this tells you is that if the mass, nanoparticle, people talk a lot about nanotechnology or safe design of nanotechnology. So what that means is that you're coming up with new materials, but what is the health effect of the stuff? And so here, of course, there is a leap that you have to take between, this MD and the health effect. But definitely there is they highlight the different tendency of shape, chemical composition, and how they affect the translocation into the membrane. So something that is more related to area, exposed, or even how hydrophobic, hydrophilic is, are interesting parameters if you, for example, want to talk about the health effect or how you characterize a nanoparticle in terms of toxicity, for example. And so I'm going to conclude with this last slide. I don't know why I chose this, but I said so this is a light micrograph of a section of a human lung. And so what you see basically is the black material. So there's a nanoparticle and soot that we've been talking up to this point. So what they found, there's an aggregate of ambient carbon ultrafine particles inside the lungs. So this is telling you something about the fact that we breathe this material and also we accumulate in the body. The second one on the bottom is a study that was done at the University of Michigan, Professor Filbert, School of Public Health. And what they did is they had a polymer nanoparticle, so the size of 60 nanometers, so a little bit bigger, more the cluster that we have seen those. And over time, they were looking at the excretion time. So what this tells you is that how the concentration of a nanoparticle goes down if you, after hours, you check, the feces, the urine, and the blood. So initially you have some concentration, and eventually the body gets rid of that. Well, the message from both those results is that somehow we are exposed to these nanomaterials, but the body is not able to get rid of that completely. So the graph below shows that even no matter how long you wait here, you are always left with some kind of concentration in the body. Somehow we don't get rid of this material. And so with this, maybe I will conclude with this, saying that one of the reasons why I started doing all those molecular simulations were more to find answers to maybe a bigger problem. There was some part of combustion, but was also more this biological stuff. And nanoparticles and the health effect, or nanomaterials and safe design. I think this is all very interesting and something that I think at this point we want to take into consideration, and maybe, you know, again, you might not be interested directly in biology, but if you are considering as avenue, as a broader view of your tools, something that is definitely on the horizon that you want to, you know, study or get interested in. So I'm going to stop here. And then, guys, if you have a question, I'll be happy to answer any questions. And also, I don't have all the pictures, but I want to, first, before I conclude, I want to acknowledge all the people in my groups that without them, none of this will be possible. So a big thank you to all my students, postdoc, research scientists that I had over the last years. Thank you.
Which range, for example, for blood, which range of osmotic pressure is being used? Because we have changes in concentration, right?
Yes.
So the transfer for the material for blood, for example, between the membrane size would be the main effective factor of the osmotic pressure, right?
The main effect of this graph is that basically you measure the concentration of those nanoparticles at different times.
The different concentration between size of membrane comes from the difference of osmotic pressure, right? The difference in concentration, because the transfer, the parameter that
I'm not transferring from
blood to transfer from side to side, the change in concentration, right?
You're doing, so you do, okay. So no, you're not transferring from one to the other. You're just sampling, you know, the blood in one thing, and you see how the body is getting rid of this concentration, basically. So you're not transferring. Oh, okay. Should I turn it off now?
No, it's fine, but you were standing next to the wall.
Oh.
It was just reverberating.
Okay, okay, that's okay. Yeah, so I was not talking about the transferring. This is more like a level that you measure in, you know, if you draw your blood in that moment, you see that still there is some concentration of nanoparticles in the system. The only message of this result is basically that somehow the body still retains some of the nanoparticles. And the one on top is the lung. So there is different way in which the nanoparticles get into your system. So one is breathing, the other one is also in the skin sometimes. So different avenues that we have that. And I guess also, maybe one question that one of you asked me, what I'm trying to, so the question was more about, you know, what we should do next. that's a you know, a great question. First of all, I think it was mentioned also today as in the career panel, try to find something that you like. And if you're lucky, you can study what you like. That's the first thing. I cannot answer directly the question, no, even DNS, MD is better. That's not my job to do that. That was one of the questions I had. But I like this, of course. But I think you need to find what you like more than anything else. So I don't know if I'm there to answer, but yeah. And again, this was meant just to give you an overview of the things that you could do with these tools. And Dr. Chan is coming tomorrow, so she will give you a great overview, also the DNS and the potential for those things. Yes?
So in this plot right here, it seems that the concentration of nanoparticles is going up. Is that like an experimental?
Yeah, that's another. I think the main point is that it doesn't go to zero. Okay. And there's an error bar on that, yes, I think for sure. Okay. Thank you, everybody, for being here.